Welcome everybody, and I'm sure there's more joining as I speak. This is the second session of the autumn series. I think this is our 12th series, the series on light. Very much welcome today our speaker, Richard Weller from the University of Edinburgh, who will be introduced by Dr. Alix Bailie. Now, Alix is a photophysical chemist undertaking a Guy Foundation funded postdoc at the Central Laser Facility at Harwell Research Campus under the guidance of Stan Botchway. And she works on fluorescence and phosphorescence imaging microscopy and super resolution microscopy in cells. So Alix, if you'd like to share your slides and give a brief introduction to our speaker.
Thank you very much for your introduction there, Geoffrey. It's my pleasure today to be introducing the topic of the health effects of sunlight, UV and blue light. The John Denver song says that sunshine on my shoulders makes me happy, and some people feel that sunshine fills their soul. But in the modern era, more and more people tell us to hide away from the sun, to use sunscreens. So what really is the effect of sunlight on the human system? The first part of that question is really, what's in a sunbeam? So the vast range of light that comes to us from the sun spans many hundreds and thousands of nanometers, as you can see from this image that you might remember from last time's talk. The UK Health Security Agency, as of 2025, recommends that we avoid this sunlight between 11 a.m. and 3 p.m. They recommend that we use an SPF of 30+ with 4-star UVA protection between March down to October. And they even say that sunburn and even a tan is a sign of overexposure to UV, but they do admit that some UV is healthy because it helps our body. So this is the widely regarded vision in the UK of how we should treat the sun, but sometimes this starts to fall down. For instance, in the news recently in Australia, there's been a massive sunscreen scandal where these SPFs have been revealed to not be 20, 30 or 40, but to be more like 4 or 6. So we've been informed that we should approach the sun in a certain way, but the industry isn't prepared to treat us properly in this way. So how should we be behaving around the sun? In the lab, the sun can also be used as a non-chemical antimicrobial. Specific wavelengths such as the UV, blue, and the near IR are used at higher intensities than we might find in the sunlight as ways to treat bacteria, fungus, and microbiomes. However, we don't have any long-term understanding of these intense wavelengths on human skin, and it seems like there's more to unravel from the sunlight itself as well. So my pleasure today is to introduce Professor Richard Weller, who is a qualified and practicing medical doctor, consultant dermatologist. He completed his initial training at St Thomas's Hospital, University of London, and then carried out research roles in Australia, Dusseldorf, Pittsburgh, and now back to Scotland, where he's a dermatology lead for NHS Scotland. Several years ago now, he presented a very interesting TEDx talk on why are Scots so sick? And hopefully he'll elaborate on some of the areas explored there that maybe we should start seeing the sunlight in a different light. So without further ado, over to you, Professor.
Alix, thank you for that nice introduction. And it's really lovely to be talking to this audience. Really interesting looking at the different aspects of sunlight, because my work is a mixture of epidemiology and also kind of mechanistic research. And of course, the epidemiology says sunlight. You can't really narrow it down much more than that. And mechanistically, you go down to the individual wavelengths. And I'm going to talk about UV. And I think where we start from, a lot of it is determined by the prejudices we approach sunlight with. So as a dermatologist, I started looking at UV because you're brought up that UV is evil and you will die. And that's what dermatologists are brought up with. And so I started looking at UV, and that's where I am. But of course, there's then visible, blue, near infrared, there's a whole world more to it. And it's fascinating how these very different specialties, medical specialties, are coming in from different angles, and a lot of it based on where you started from. So This is Edinburgh with, I think, a period of no UV, setting sun, so a UV-free zone, as Edinburgh is for much of the year. This is actually where I work. I'm based out here in the Institute for Regeneration and Repair on the new campus on the edge of town. And I'm going to talk today a bit of mechanistic work, sunlight and nitric oxide, a bit of epidemiology, sunlight and all-cause mortality. I'm going to talk about some work we're currently doing about some new immune mechanisms by which sunlight activates its beneficial effects, which we're studying. And also I'm touching on the evolution of skin color, which again is another important and interesting area. And I'm particularly really concentrating on UV. So, changing attitude to sunlight. Our attitudes, of course, to sunlight have really changed dramatically in the last 100 years. I was invited to give the opening talk for the Swiss Dermatology Association, I think 2 years ago, in Lausanne. And as I pointed out to them, if I'd been there 100 years ago, I would have gone there as a disciple. You know, Rollier started heliotherapy, UV treatment effectively of tuberculosis. And that was really a central part of medicine then. Here I am coming back 100 years later to remind them of what they used to do. So, you know, you went to Davos or Lausanne for getting heliotherapy for tuberculosis. Or if you lived in Britain, this is Bristol. Children were treated for rickets with UV. If you lived in Edinburgh, this little hut on a railway turntable was actually in the grounds of the hospital where I work, in the Western General Hospital. And we couldn't afford to send our patients to Davos. Used to put them in a little hut in the hospital grounds and turn it to face the sun. Or if you lived in Glasgow, you could put your child in a cage outside the window, and that was to prevent rickets. So that was then. And this is a standard dermatologist on holiday on the beach today. An entirely different approach to sunlight. And of course, you know, what's driven this completely different approach to sunlight? And it's fear of skin cancer. So this really dominates the discourse about UV in much of the world today, driven by dermatologists' concerns about UV and skin cancer. But where does UV rank in terms of overall risks to health? So the World Health Organization every 5 years draws up a list of the leading risk factors for premature death and disease. So DALY's, disability adjusted life years, is a composite score of early death and disease. They basically take the Japanese, who live the longest in the world, and see how close the rest of us get to being Japanese. But you can rank risk factors that lead to DALYs, and the leading risk factor in the world today is high blood pressure, and that's because high blood pressure underlies strokes and heart attacks, and they're the two big causes of death, and high blood pressure is their major risk factor leading to those causes. And in fact, high blood pressure has now overtaken cigarette smoking. And what is interesting about blood pressure at population level is on the left, this graph on the left, each of those dots is a different country in the world. And the y-axis is systolic blood pressure, and the x-axis is latitude. So the equator in the middle, northern hemisphere to the right, southern hemisphere to the left. And this data comes from 1980. And the point about that is 1980 is before the invention of modern, very effective antihypertensive agents, because high blood pressure is such a health problem that in the rich world, we now have a strong focus, a preventive medicine focus on controlling blood pressure. And actually, blood pressure has dropped in the rich world in the last 40 years, but that's due to pharmacological intervention. But before that, you can see very clearly that the further a country is from the equator, the higher population blood pressure is. Now, it could be that at the North Pole and the South Pole, there is a hobgoblin and his twin on the other pole squirting out a gas that causes hypertension. That's entirely consistent with the data. However, if you look at blood pressure by month of the year, on the right is a graph from a paper which I think I learned at medical school or as a junior doctor, 2 different treatments for hypertension. But when you analyze blood pressure by the month of the year in which the blood pressure was taken, we see there's a huge seasonal variation in blood pressure in the UK. So in summer, blood pressure is significantly lower than winter. And in fact, the effect of season is as great as the effect of a drug in lowering blood pressure. So what this might suggest to you is that sunnier countries, or within a country, sunnier times of the year correlate with lower blood pressure. And here we are again, the more UV that falls, the lower cardiovascular disease incidence, which is a sequela of high blood pressure. So that's observational data. The next bit of observational data is really looking at vitamin D. So UVB formation is responsible for vitamin D synthesis in man. So blood vitamin D levels are a great indicator, a biomarker for how much sunlight and specifically UVB you've had. And we know that people with lower vitamin D, measured vitamin D levels, are more likely to have hypertension, more likely to have cardiovascular disease, more likely to die of stroke, and in fact, more likely to die of any cause whatsoever. So that's observational data. The problem is when you carry out interventional studies, when you enter people into randomized, placebo-controlled, double-blinded trials of vitamin D supplementation, it does pretty much nothing. So this is a meta-analysis, a combined analysis of multiple trials of vitamin D supplementation, published in the Lancet, 12 years ago now. Giving people vitamin D has no effect on myocardial infarctions, no effect on strokes, and no effects on cancer deaths. And the biggest study of all was completed about 5 or 6 years ago in America. So this was a study called the VITAL study, funded by the NIH in America. 25,000 Americans randomized, half to get vitamin D supplements for 5 years, half to get placebo. The results have been published largely in the New England Journal, and the results are broadly negative, 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 negative. Some possible effects on autoimmune disease. So that was 25,000 people for 5 years. The Australians ran a smaller study, 21,000 people for 5 years. Half get vitamin D, half get placebo. Results are negative, 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 negative. So it's not the vitamin D. And this is an editorial from the New England Journal from 2022, 4 years ago now, saying, adding those findings to previous reports, etc., people should stop taking vitamin D supplements to prevent major diseases or extend life. So clinical trials confirm it's not the vitamin D. So vitamin D is a biomarker for sunlight exposure, and it prevents rickets in children, to some benefits, prevention of rickets being the main one. So we have sunlight here. Over here we have diseases, rickets, and then everything else. If you're a male living in Scotland, you are 30% more likely to drop dead in a week in January than a week in August. Huge, huge. We've just passed the equinox, heading the wrong way. Time of year when I start to get a bit antsy, summer's fine. Am I going to survive another winter? The data is pretty serious. We know that vitamin D accounts for rickets, prevention of rickets, but what about the rest? And that's really the question that we're facing now. So this is observational data, and the vitamin D debunked by trial data, limited benefits. So I'm going to move on now to the first bit of mechanism, and I'm going to talk about nitric oxide. And I'm going to start really talking about nitrogen oxides, nitrate NO3, nitrite NO2, nitrosophiles. So about, well, first of all, I described years back, 30 years ago, almost, this reduction of NO on the skin surface by bacterial reduction of nitrate. I then got diverted from that and I went off to the States and I went to Germany, spent time abroad. At a time when I was still dermatologically speaking a good boy, I'm now dermatologically speaking very much a bad boy, but I was still in dermatologist terms a good boy, and I thought that all of this was something to do with skin cancer because UV is terrible stuff and causes skin cancer. Did lots of cell culture work in Germany, mouse models in America, came back, couldn't reproduce my mouse work in man when I came back. I discussed this in a TED talk I gave. But problem solving those experiments, we showed that the skin contains large stores of nitrogen oxides, nitrate, nitrite, nitrosophiles. And at the time, I did not know what that meant. It was thought that nitrate was an inert end product of nitric oxide oxidation. It was thought it did nothing and was peed out. But I found all this stuff in the skin for no obvious cause, and I sent it off to the JID, which was the top dermatology journal. I sent the usual cover letter, groundbreaking science, you're so lucky to get this, do brilliant stuff, you should really take it, whilst thinking to myself, I don't know what this means, I have no idea, I think this is rather odd. Anyway, they accepted the paper, much to my surprise, because I wouldn't have done so. And it's actually turned out to be a really important paper. So I'm going to move on now to how nitric oxide is synthesized. So nitric oxide, which we're talking about, was first shown by Bob Furchgott in America. This simple diatomic molecule was shown to be released from arginine by nitric oxide synthase, this enzyme. And this was then, and what he then showed was that he got the Nobel Prize for this work later on, was he showed that NO is cleaved from arginine by a nitric oxide synthase, dilates blood vessels, lowers blood pressure. And of course, what was exciting and unusual was that NO is a very simple diatomic molecule. You know, it has two atoms. And previously, when we think about chemical messengers, signals being passed from one cell to another, we've thought of proteins with complex 3D structures, you know, acetylcholine binding to an acetylcholine receptor, insulin binding to an insulin receptors, complex 3D shapes binding to complex 3D receptors. And that complexity gave specificity because a complex protein only, an agonist only matches to a very specific receptor. And here you have this incredibly simple, rather non-specific agonist. And how do you, first of all, focus that signal? And secondly, how do you turn it off? You know, an agonist unbinds from a receptor, but what happens here? So what was realized was that actually nitric oxide has a very short half-life, you know, just a couple of seconds in biological tissues. It is then oxidized rapidly to nitrite, NO2, which has a half-life of minutes to hours. And that is then oxidized to nitrate, NO3. And the idea was that nitrate is biologically inert and you pee it out. So that was the story. Exciting stuff, new science, Nobel Prize for Medicine in 1998. So what's that, 28 years ago, yesterday, wasn't it, when they award the Nobel Prize? So that was the Nobel Prize winning stuff. I was at this point working in Aberdeen as a dermatology registrar, and this inspirational character, really my mentor who got me into research, fantastic guy, Ben Benjamin, who was a young clinical pharmacologist up in Aberdeen, and he showed a second pathway by which nitric oxide can be produced. He showed that nitrates can actually be acted on by bacteria. So bacteria contain nitrate reductase, And those can reduce nitrate to nitrite. And this he described occurring in saliva on the tongue. So your salivary glands concentrate nitrate from your plasma into the saliva. And your tongue is covered in bacteria, and these, particularly the back of the tongue, and these bacteria reduce that nitrate to nitrite, which you then swallow to your stomach. And in the acid conditions of the stomach, that NO is released and goes into the circulation. So that was in 1995. Before the Nobel Prize, NO was very much the hot topic, very exciting. And it was a kind of rule. It was a rule of biology that there were no mammalian nitrate reductases. The old way you used to test for bacteria in the 1950s, actually, was you saw if there was nitrate reducing activity, and that was a biological test for the presence of staph bacteria. So microbiology labs did a nitrate reductase thing, and they said, oh, nitrate reductase, it's a gram-positive bacterium. But the next person to jump on the scene was John Lundberg at the Karolinska. And John showed that actually there is a bacterial nitrate reductase. He first of all showed it in rats, and then he showed it in Swedish athletes in whom he did muscle biopsies. And he showed that actually there is a mammalian bacterial nitrate reductase. And he then showed that if you take a high nitrate diet, classically beetroot, that you increase circulating NO via a mammalian bacterial nitrate reductase acting on the oral nitrate, on the dietary nitrate. And that not only releases NO to the circulation, it also improves athletic efficiency, because what he then showed was that you have more efficient generation of ATP by mitochondria in the presence of oral nitrate that reduces the electron leakiness of those electron transport chains. And in fact, so much so that oral nitrate became something athletes took. And before the London Olympics, John had to go up to the IOC, the International Olympic Committee, medical committee, giving evidence as to whether beetroot should be banned as a performance-enhancing vegetable. And the IOC passed, so beetroot, so apparently the urinals in London were running pink, and endurance athletes to this day now take a high nitrate beetroot juice. So that's two pathways for NO. And then, and importantly to this story, a third pathway was produced. So the nitric oxide world at that stage was divided into two fairly, can I say viciously, fairly antagonistic parties, all based on what levels of NO they thought were biologically active. So there was the predominantly European and half of American groups who were finding one level of NO accounting for things. And then there was an American group, and in their experiments, they were producing figures for NO in biological samples 1,000 times higher than the other groups. So you had these two very different groups, both groups publishing in good journals, the high NO bunch publishing Science and Nature, but their figures, their numbers did not add up. So Martin, another friend and collaborator, he set out to do a very dry and boring methods paper. And he set out because it turned out that the two opposing groups measured nitric oxide in different ways. So John's group, Ben's group, my group, Salvador Moncada, the Nobel Prize group, we all measured nitric oxide via a technique called chemiluminescence. You mix ozone with NO, it briefly combines, and then it releases light. So that's the chemiluminescence-based method. The other method of measuring NO involved flash lamp photolysis. So you shine a brief, very intense burst of UV at your biological sample. And by that UV absorption, you could infer the NO level by the pattern of NO absorption. And that group were getting much higher levels of NO. So what Martin showed in this methods paper that nobody read was that when you shine UV at nitrate in the presence of thiols, SH groups, you get a photochemical reduction of nitrate to nitrite. So the reason that the high NO group were getting high NO figures is because their measurement method was releasing NO. So here you have this dry paper, which is all about how you measure things, and whoever reads methods papers, I don't, nobody does, stamp collectors do. So that's where we were, and I was very much involved with this nitrate reductase pathway, until we had there was a kind of work, there's an NO group, and we meet every year. And the meeting in 2008 was in Bregent's. So Bregent's is an opera festival on the stage on the lake. If any of you have seen the James Bond movie Quantum of Solace, there's a chase scene on the opera stage here. And so my wife and I went out there. We went to the opera. It was Tosca. And then the opera festival finished, and the opera house was cheap to rent. So being scientists, the NO group needed a cheap venue, and we rented the opera house post-festival. And as they were taking the stage down. And at the bar, after a day of science, I was saying to Martin and my friends around the table, I've discovered these huge nitrate stores in the skin. I have no idea what it means. It seems I've managed to publish it, can't understand why. What does it mean? And Martin said, I've written this paper 3 years ago that nobody has read. We show that UV releases NO from nitrate in the presence of thiols. And I went, oh my God, UV hits the skin. The skin's got lots of thiols. It's got all these keratins containing thiols in the skin. And it's got lots of nitrate. You've got the 3 ingredients that make NO. And talking over the bar, I could remember from medical school days that in sunny times of year, blood pressure is lower. Could this account for those seasonal variations in blood pressure? So those of us standing around the bar, so Martin, Victoria Kolbachhofen, my old boss from Dusseldorf, Donald, my PhD student, John Lundberg, nitrate reducing, the blood pressure treating beetroots. All my colleagues got together and we wrote what I've always called my lamppost paper. We wanted to mark the territory. This is our idea, we've had it. So we quickly published this hypothesis paper in the European Heart Journal saying we think this might be happening. We think that sunlight hits the skin. We think that it photo-releases these nitric oxide stores to the circulation, which lowers blood pressure. And we then set out to try and confirm it. So Christoph Sustrek was doing this in Dusseldorf, and I was doing this in Edinburgh. Christoph got there before me for the first paper, but I think did a slightly better controlled paper. The ethics process is so mind-numbingly slow in Britain. It took me 3 years to do this because the bureaucratic inertia around doing human studies is overwhelming. So simple idea, it took 3 years to do the experiment. So with Donald, my PhD student, we first of all, we took skin. These are redundant samples from plastic surgery, and you shine UV at skin, and this green fluorochrome is an NO marker. And we show that the more UV you shine at skin, the more NO is released. But when you sham irradiate the skin, so we irradiate it, but it's covered with foil, so the temperature rises in the same way as the active skin, but no rays hit the skin. So no release of NO in the sham irradiated, and dose-dependent release of NO in the irradiated. And we chose UVA because I wanted to show that this was a vitamin D independent process. So it's UVB that makes vitamin D. So I chose UVA because I know whatever you do, the vitamin D industry will come back and say, oh, it's vitamin D, take a tablet. So we were clearly trying to show a difference from vitamin D. So UV releases NO in the skin. We then did these forearm plethysmography experiments. So you take human volunteers, you put a blood pressure cuff around the upper arm, and you inflate the blood pressure cuff to above venous but below arterial pressure. So blood can flow into the arm but not out. You put a strain gauge around the forearm, and by the amount the strain gauge goes up, it tells you how much blood has flowed in. And of course, arterial vasodilation allows more blood to flow in. So that strain gauge is telling you you've got arterial vasodilatation. You then cannulate the brachial artery, and into the arterial vasculature in the forearm, you can infuse a drug of your choice. So we infused this drug, LNMMA, which turns off the nitric oxide synthase pathway, the Nobel Prize-winning pathway, but it doesn't turn off nitrate reduction. The technique of forearm plethysmography was developed by three registrars, three trainee clinical pharmacologists at George's in about 1980. So one was Ben Benjamin, my mentor, who taught it to me. The other was David Webb, who's professor of clinical pharmacology here in Edinburgh. And the third of those three registrars was a chap called Patrick Vallance. And apparently he went into the civil service. Don't know what happened to him. For those who don't follow, he's chief scientist. Anyway, so those three people developed it. They were three registrars together. And Patrick has sunk into obscurity, but the other two had very good careers. So we did that, and we then shone UV at the arm. And we either shone UV at the arm, so rays hit it, or we shone UV at the arm, but it was covered with a foil blanket, so the temperature rises, but UV doesn't hit the skin. And what we showed was that when you irradiate the forearm, it causes vasodilation. So ultraviolet in man is an arterial vasodilator, and the sham arm, there was no vasodilatation. So UV in man is a direct vasodilator, and blood pressure is a function of total peripheral resistance and cardiac output. So you multiply what the cardiac output is by the peripheral resistance, and the more constricted vessels are, the greater the resistance. And the function of those two gives you blood pressure. So we then stood people in UV cabinets, or we laid people under full-length UV lamps, and we showed that shining UV at people lowers blood pressure. The sham irradiation covered the foil blanket so that the temperature goes up but not the rays. There's a fall in blood pressure while the lamps are on because you get warm, but as soon as the lamps are up, the sham irradiation returns to normal, but the active irradiated stays down. Oh, and then you have a rise in circulating nitric oxide in the irradiated group and a fall in nitrate. So sunlight hits the skin, releases NO, which moves into the circulation, which dilates arteries, which lowers blood pressure, so you don't get a heart attack or stroke and you live longer. That's great. So that was super. Of absolutely zero interest to dermatologists, not interested at all. Couldn't be, they really don't care. So we had Harold Varmus, who, so I presented that first. We had in Edinburgh the World Congress of Dermatology, the biggest academic dermatology meeting in the world, was held in Edinburgh in 2014 when I'd just done this. And I thought, wow, this is huge. This is really big. And I submit it to the meeting, think plenary, they'll all be hanging on every word. There'll be 1,000 people hanging on every word. To my horror, I was given a poster. So it's just around the corner from me. It was here in Edinburgh. Harold Varmus was coming to Edinburgh University at the very same time to the university. So Harold Varmus was head of the National Cancer Institute, Nobel Prize for Medicine, an absolute god of biomedical research. And a friend rang me up saying he's really keen on going out and doing things. He wants to walk up a Munro. Can you join us? Just a car of us, 3 of us, 4 of us, going for a day out with Harold Varmus going to stomp up a Munro. And I thought, oh my God, you know, Nobel laureate for a day, head of the NCI, fantastic. But it was the day my poster was going to be up. And I said, oh, but I have this incredibly important piece of work. I'm going to be flooded by people. He rang me the day before saying, look, do you know who Harold Varmus is? And I said, no, can't go. And I sat by my poster and I had one visitor. And what it taught, so I don't go to dermatology meetings much nowadays, you know, just no interest. I go if I'm invited, but otherwise I don't present work. I don't submit work there because there is no interest in this from dermatologists. However, a lot of interest from general medical physicians, from internal medicine. And I was approached by the Renal Research Institute. So the Renal Research Institute is the research arm of Fresenius. So Fresenius make dialysis machines. It's an Austrian or German company, makes most of the dialysis machines in the world. And they run 3/4 of the dialysis units in America. And blood pressure control is a real problem for dialysis patients because they don't have kidneys. Giving them drugs is difficult because they're going to be renally cleared. And so they're very interested in trying to optimize care for their patients. And they have 340,000 patients they're dialyzing in America in 2,000 different centers. And we realized we could actually get in and the great thing about dialysis patients is they have their blood pressure measured 3 times a week when they get dialyzed. Week in, week out, year in, year out. So you have over 1/3 of a million patients, 3 blood pressure measurements a week over, we did 3 years, and in 2,000 different dialysis centers. And the point about these different dialysis centers is that at each one you have different temperature, you have different UV, you have a different spectrum of UV, and you have a different energy of UV. You know, as the year changes, depending on your latitude, on your altitude. And with satellite data for each of those centers, you can derive the amount of UV, UVA, UVB split, temperature, and you can cross-reference that to the blood pressure in those patients. And you then have data on skin color. So they had 100,000 Black Americans and 200,000 white Americans. So we got in there and we showed, so what we showed was that about half of seasonal variation in blood pressure is due to temperature. So part of that fall in blood pressure in summer is due to temperature, but only about half of it. The other half is independent of temperature. It's caused by UV alone. And we also know it's not caused by vitamin D because we've done the trials. Because for the last 100 years, dermatologists have said, see no evil, speak no evil, hear no evil. Sunlight, blood pressure, no, it's temperature, it's vitamin D. Okay, so it's neither of the above, or it's only partially temperature. So what we could show was, although we had used UVA for my experiments to say it's not vitamin D, actually the dose response curves showed a bigger fall in blood pressure with UVB than UVA, although both had an effect. The other important one was that Black Americans had less of a fall in blood pressure for the same rise in UV as white Americans. Now, I'm moving on to this later, but this is not surprising. Skin color is an evolutionary adaptation to different amounts of sunlight. And of course, the real problem here is that Black Americans have and Black Britons have higher blood pressure than white Americans and white Britons. And with that, they have higher rates of strokes and heart attacks, despite which they are told to avoid sunlight. But I have to say, it's madness. Okay, so here we are, further confirmation at population level of the effects of sunlight on blood pressure. And what does this mean? So we have this UV-driven fall in blood pressure. So in the UK, seasonal variation in blood pressure, the winter to summer variation in blood pressure in the UK is about 6 millimeters of mercury systolic, 3 diastolic. So about 5.6 over 3.3. Now, about half that variation is due to temperature, as our data from America shows, but half of it is due to UV alone. What we know, this is a meta-analysis published in The Lancet of multiple trials of pharmacological treatments for hypertension. And what we see is the more you lower blood pressure on the x-axis, dose-dependently and linearly, the more you lower the risk of cardiovascular events and deaths on the y-axis. So lowering blood pressure, however much or from whatever starting point, reduces risks of cardiovascular events. So we have a seasonal variation of 6 millimeters. So summer blood pressure, summer sunlight lowers blood pressure by 6 millimeters of mercury, which you would anticipate would cause a 20% reduction in cardiovascular events. And indeed, in winter, other way round, deaths from heart disease are 23% higher than in summer, exactly what would be predicted by that variation in blood pressure. So look, about 10%, so cardiovascular deaths, you can say about 10% of cardiovascular deaths in winter would be caused by that lack of sunlight. So say, as a thought experiment, that dermatologists achieved their goal and they completely eradicated deaths from melanoma in Britain. They wouldn't, by the way, at all, but for the sake of the argument, let's say so, by getting everybody to live at winter levels of sunshine all the time. And with that, you had a 10% rise in cardiovascular deaths, which is what you'd predict. So you spare 2,500 melanoma deaths, but you cause an extra 10% of 170,000 cardiovascular deaths. 10% of 170,000 is more than 2,500. So just as a thought experiment, which I did back then, this would suggest that reducing sunlight to winter levels is perhaps not a good thing. So, talk so far. Hypertension is the biggest killer globally. Nitric oxide stores in the skin mobilized by UV lower blood pressure in man. And now I'm going to talk about some epidemiology about all-cause mortality. So I've set the idea that maybe lowering sunlight exposure would have a bigger adverse effect than good effect. But of course, the way to look at this is to look at all-cause mortality, death from any cause. So, do the benefits of sunlight, blood pressure reduction, outweigh the harms from sunlight, increase in skin cancer, or is it the other way around? So you look at all-cause mortality. So the first person to do this was Pelle Lindqvist, who is a wonderful obstetrician in Sweden, who I've got to know. Pelle is actually, he was interested in preventing blood clots in pregnant women, and he was looking at this for that reason. So in Sweden, they have set up a prospective, a forward-looking study on risk factors for melanoma. And it was set up in 1990, the melanoma in southern Sweden study. So they recruited about a quarter of middle-aged women in southern Sweden at that stage. And at that period, 1990, Sweden had no immigration, so it was a purely white population because skin color determines response to UV, so you have to consider it. So these 30,000 Swedish women sunbathe in summer? Do you sunbathe in winter? Do you go on foreign holidays? Do you go on tanning beds? Now, obviously, there's lots of confounders here. If you go on foreign holidays, you're richer, so you're probably better educated, so you're less likely to smoke, etc., etc. But he adjusted for all of those. He adjusted for social factors like income, education, marital status. In Sweden, everybody's tax return is published openly on the same day each year. If you want to find out what your boss earns, or the prime minister, or your next door neighbor, you can look it up. So they threw that in. They threw in health factors like exercise, smoking, alcohol, pregnancies, BMI. They threw in all of these factors. And they went back 25 years later to find out what had happened. So you had a sunlight exposure score of 0 to 4, 4 being the highest. what were their risks of being dead after adjusting for these confounders? So those Swedes, those naughty Swedes who enjoyed their dermatologist and got as much sun as they could, were half as likely to be dead as those goody two-shoes loved by their dermatologist, looked great, no solar, no photo aging, but dead. Half. The sized smokers who got lots of sunlight had the same mortality as people who avoided the sun and didn't smoke. That is the size. Smoking is the worst thing you can do for your health. It's always held up as the worst thing you can do. That is the size of the effect. The biggest reduction was in cardiovascular deaths. And in fact, there was an apparent increase in cancer mortality in the sun seekers. But the reason for this was they had such a reduction in cardiovascular deaths that, that there was an apparent rise in cancer deaths, but it wasn't real. It's just that they were not dying of other things. Overall, the mortality greatly reduced in those people that got the most sunlight. The other interesting thing, we're going to come on to skin color later, is he looked at the Swedes who got the least sunlight, so those Swedes who avoided the sunlight. So red hair, the palest phenotype that humans have is red hair. So when you have red hair, you have a different type of melanin. It's a genetic variation that leads to this incredibly pale skin and red hair. And redheads are found around the fringes of Northwest Europe, the furthest away we've got from Africa, which is where we all come from. And what Pelly showed was that redheads, when you look at Swedes who avoid the sun, redheads have a survival advantage over white non-redheads. But when you look at Swedes who get average or high amounts of sunlight, redheads lose that survival advantage. So the palest skin of all is an evolutionary adaptation to lack of sunlight. We need it. And if there isn't any sunlight, if you live in northern Sweden and don't see much sun, you're really struggling. So the evolutionary fitness favors this very pale phenotype. So that was Pelly. Sweden's a bijou little country. We thought we'd throw some real data at it. So we went to the UK Biobank. So the UK Biobank is this amazing resource. 500,000 people in Britain, I was one of them, who were recruited over a 3-year period finishing 15 years ago. They went round 14 different centres in the UK. It took 3 years to recruit all these people. There was about a 3-hour assessment when you went up to it. Huge, great health questionnaires, masses of measurements taken, bloods taken, really wonderful stuff. One of the great things the Biobank did was they banked blood from all of those participants, knowing that technologies would be invented in future years that could be used for examining those bloods. So already we've got whole genome sequencing, which is suddenly incredibly cheap. Proteomics in the last year, a technique only invented 5 years ago, gives you amazing proteomic data. So it was really well thought out. You bank bloods against technologies that are going to be invented and have been, and give us even more data. And we wanted to look at the effects of sunlight exposure on all-cause mortality. So in the questionnaires, there were in the data set, there are 2 bits of information we use that give us an indicator about sunlight exposure. The first thing was we looked at sunbed users. Now, we've called them sun seekers so as not to frighten dermatologists. This is because we know that sunbed users like sunlight. We know that they sunbathe more. It's a behavioral marker. Sunbed users actively seek out the sun. So that was our behavioral marker. The other was a geographical marker. We looked at how far north and south people live. And using satellite data, we have 10 by 10 meter square patches over the whole of the UK, and we can calculate how much UV falls on every single 10 by 10 meter patch of the UV for the UK. So for every person in the biobank, we know where they live, their lat and long, and we can tell how sunny it is where they live. Now, you then, of course, have to adjust for confounders. Oh, so here we are. So we analysed this lot 12 and a half years after recruitment, 28,000 deaths. I mean, it's not many, but the data gets better with time. You know, the longer you wait, the more they die, the better the data gets. So the great thing about being an epidemiologist is the data only improves. I work with epidemiologists and they sit there drinking cups of coffee and I say, you're being bone idle, you should do some work. And they say, I am working. You know, I'm waiting. More people will die. The data gets better. So my next life, I shall be an epidemiologist. And of course, the important thing is you have to adjust for confounders. So a confounder is a factor independently associated with the exposure, you know, sunbed use, living further south, and the outcome, death. So these are the confounders we adjust for. Now, the important thing in this experimental design is the confounders move differently for our different exposures. So the problem with observational studies, the weakness of observational studies is that there might be an unmeasured confounder accounting for your outcomes. So say the real secret to a long life lies in painting your toenails green while singing in the bath and doing the Times crossword. None of us expected that those three things are actually the secret to a long life. Now, with our two different exposures, the confounders moved in very different ways. So sunbed users are, you know, younger, more female, more Mancunian, more likely to smoke, less educated. Whereas people who live further south are younger, more female, more educated, less likely to smoke. The point is the confounders move in very different ways, yet both of them are indicators for sunlight exposure. And because the confounders move in very different ways, it is incredibly unlikely that humming in the bath, doing the Times crossword, painting your toenails green should be found equally in both groups when all of the other confounders move differently. So it's called triangulation, and it's a way in observational studies you try and reduce the risk of unmeasured confounders. So a really well-designed study. And the next thing we did was we used vitamin D level. So remember, serum vitamin D is a biomarker for sunlight exposure. And our sun seekers had higher vitamin D levels than our non-sun seekers. So we know that our behavioral marker is a good marker for sunlight exposure. So what happened 12 and a half years after recruitment finished? So sun seekers, after adjusting confounders, were 15% less likely to be dead from any cause. In particular, a 23% reduction in cardiovascular deaths, 14% reduction in cancer deaths, and other deaths, things like multiple sclerosis, diabetes, you know, the rest, 12%. Their life expectancy after 12 and a half years was 50 days longer. It may not sound much. It's longer than some modern conservative premierships. You know, 50 days after 12 and a half years. Now, whether it's 100 days after 25 years, whether it's 100, I don't know, but significantly increased lifespan. So dermatologists instantly say, what about melanoma? So yes, there was an increased incidence in melanoma. Numbers are small. No increase in mortality, but numbers are small. All other cancers, a reduction in mortality. These are the groupings of the major cancers, all of the other cancers. So that the big reduction, cardiovascular deaths, but the other cancer deaths also all reduced. So that's our first indicator of sunlight exposure. What about the geographical one? So here we are, as an example, the further south people live, Glasgow, Nottingham, Truro, I've given as an examples because that's the top and the bottom and the midpoint. We have increasing amounts of UV over the year fall in those three different sites. And dose dependently, the further south people live, the higher their vitamin D levels. So again, we know our geographical measure is a realistic measure for how much sunlight people get in their lives. So what happens to people that get more sunlight exposure after adjusting for all of these confounders? So the difference between Glasgow and Truro, 12% lower all-cause, 19% cardiovascular, 12% cancer. Doesn't quite hit significance, the rest. 300 kilometers further south, so that's Edinburgh to Manchester, is 16 days longer lifespan after 12 years. And again, Truro to Glasgow, an increased incidence, case of melanoma, no change in mortality. All other cancer mortality down. So this was published last year. For the British population, the more sunlight they get, the longer they live. We're about to submit a paper on work we've done more recently. We have gone back and we have incorporated multiple measures. So the previous study was 2 separate studies, independently looking at sunlight and all-cause mortality. Different measures, the same answer. What we've now done is we've combined all the measures into a single multi-dimensional score, which we've called the Sunbeam score. You have to have a good acronym. Sun, Behavioral and Environmental Exposure Measure. If you've got a good acronym, that's half the battle done. So we have a Sunbeam score. So we're looking at solarium use, time outdoors, more or less than 4 hours a day. Sunscreen users actually get more sunlight in the UK. Where you live. We divided people into the highest and the lowest sunny. 0 to 4. And for the 500,000 people, or 400,000 who we've used, and we've got the full data set, we can divide them up into low, medium, and high sunlight exposure cohorts. So with these numbers, that they're not evenly balanced because not many people, sadly, in the UK, and that's a problem for us, get high UV. You can then work out what proportion of disease and death is caused by sunlight or absence of sunlight when you know how people die in these cohorts. So what you do is you can compare deaths from various causes in people in the medium UV group compared to the low UV group, or high UV group compared to the low UV group. So this is what you see here. So the blue dots are comparing low with medium. The red dots are comparing high with low UV. And what you find dose dependently, the more sunlight people get, the greater the reduction in all-cause mortality, the greater the reduction in cardiovascular mortality, the greater the reduction in cancer mortality. Skin cancers, interestingly, melanoma caused by sunlight. Melanoma, the association with melanoma and sunlight is sunburn, not sunlight. People with higher vitamin D levels, we know, are less likely to be diagnosed with melanoma. We know that people with high vitamin D levels that have melanoma are less likely to die of it. Clinical trials of giving people vitamin D who have melanoma shows it has no difference on outcome. So actually there's a lot of evidence that chronic sun exposure, non-burning, reduces deaths from melanoma. Now that's not discussed by dermatologists, it's too complicated for the public. Absolutely it's known by melanoma epidemiologists, and look at this. You know, the high UV group have no, anyway. So this is a dose dependency. You cannot prove causation from observational studies. That's one of the rules of, to prove causation, you have to do interventional studies. But you can get a very strong steer to causation from observational studies. So Dolan Hill, who showed the great epidemiologist, smoking causes lung cancer, they said, things in observational studies that help you infer causality are timing. So, you know, you smoke before you get lung cancer. You don't get lung cancer before you smoke. So the timing. Mechanism. So, you know, smoking does damage to DNA. We have mechanism for sunlight. We have nitric oxide. We've got some gene regulations. And secondly, dose dependency. And we very clearly have dose dependency here. I might say we know that smoking causes lung cancer. There is no clinical trial to show that. It would be unethical to do it. We infer that smoking causes lung cancer because people smoke before they get lung cancer, because there is dose dependency. Someone smoking 20 a day is twice as likely as someone smoking 10 a day to get lung cancer. And we have mechanistic studies on what nicotine and tar does to lung cells. It's all observational, but it's absolutely robust. And when people stop smoking, their risk from dying from lung cancer goes away. So here we have dose dependency for sunlight and reduced mortality from these major causes in Britain. Next, and far more importantly, we can use this equation here to work out what percentage of skin cancer deaths are caused by sunlight, and what percentage of these other deaths are caused by inadequate sunlight. So 9% of melanoma deaths can be attributed to excess sunlight from, high sunlight exposure. However, all-cause mortality is the other way around. 6.6% of deaths from all-cause in the UK biobank. So we're talking about a middle-aged white British cohort. Maybe it doesn't apply to other cohorts. It probably doesn't apply in Australia because they get lots of sunlight there, but it applies here and it applies in Scandinavia. 6.6% of deaths all caused due to inadequate sunlight, particularly cardiovascular, 11%, also cancers, 4.6%. So what does that mean? So these are the deaths in the UK biobank. So we're now 15 years after the biobank closed. So we've got, the great news is we've got more deaths and better data. So these are the number of people who've died in the UK biobank. So 34,000 of those 400,000 have died. The big killer is cancer. So sunlight has only got a 4.6 association with cancer deaths, as opposed to cardiovascular 10%. But cancer deaths are much bigger than cardiovascular deaths now. We've stopped people smoking, we've lowered blood pressure, we've lowered cholesterol, lots of good public health measures. And we've had 342 melanoma deaths and 48 keratinocyte cancer deaths. So what you do to work out the number of deaths caused by or prevented by sunlight is you multiply these percentages by these death numbers. So in the 15 years since the biobank, so 39 people have died from melanoma, which could be attributed to too much sunlight. I can't tell which 39, but of 400,000 people, 1 in 10,000 people in the UK biobank has died of melanoma caused by sunlight. Not a lot. There's only been 1 death from keratinocyte cancer, probably because people die of keratinocyte cancer in their 90s, and there's nobody older than 75. So that's deaths attributable to excess sunlight. What about the other deaths? These are deaths attributable to insufficient sunlight. This is us in this country. This is you and me. Okay? So we have 2,982 deaths which can be attributed epidemiologically to insufficient sunlight. So if we lower sunlight exposure enough to prevent 1 death from skin cancer, it would lead to 75 excess deaths from other causes. We have got our health message catastrophically wrong, and dermatologists just don't want to know about it. So I'm going to move on to a bit of mechanistic stuff now. Taking you back, so look, I was talking to the Swiss again. We're getting a combined Zurich Edinburgh skin research club together. Here we are, Thomas Mann, the Silver Mountain. So what's really interesting, of course, is 100 years ago, UV was used to treat tuberculosis. So you had your pulmonary or respiratory tuberculosis treated by going up to heliotherapy, up to the mountains. And cutaneous tuberculosis of the skin was treated with UV to the skin, famously prize in 1904, the only dermatologist ever to receive it. And dermatologists don't know whether to applaud it because he was a dermatologist or decry it because it was advocating UV. Oh my God. So what do we do? So Finson was treating cutaneous TB with UV. Interesting, this is the light room in London Hospital. King Edward and Queen Alexandra visiting. Queen Alexandra was Danish. I do just wonder if she went to see a Danish invention thing. Anyway, none of them published decent data. I've been to look at their works. This was the era before clinical trials. They just say they got better. It's wonderful. So I thought, well, look, I wonder what actually happens. So I went back. This is unpublished kind of work we're looking at. I thought, let's look at cases, not deaths, but cases of disease using our medium and high UV exposure. And let's looking at GP records and hospital records. So look, so dose dependently, we find more sunlight, lower cases of multiple sclerosis develop. We know that multiple sclerosis is a sunlight deprivation disease. Here's all the cardiovascular things, you know, strokes, heart attacks, all the rest of it. Dose dependently, the more sunlight you get in the UK, the less likely you are to have a stroke, to have a heart attack. We know all of that. You're more likely to get photoaging. Oh dear, how worrying. I'm not a beautician. Sadly, many dermatologists are beauticians before they're doctors. I really couldn't give a monkeys. Eczema and psoriasis reduced. What about tuberculosis, I thought? I pulled this out 2 weeks ago. So look at this, dose dependently, the more sunlight people have in the UK, the less likely they are to develop UV. So even though Finson and Rollier were not doing clinical trials, they were right. Fascinating. So look, I would take the antibiotics now, but it's very interesting. If you haven't got the antibiotics, here's the best thing. So here we are, hypertension, biggest killer, all-cause mortality. So what about mechanisms? I've spoken about the nitric oxide work we've done. I'm going to give you a brief show of some of the work we're doing now. So we're now, look, this cancer mechanism is really interesting. So what we do is in winter, we take, so the great thing about living in Scotland is we have a period of 6 months a year when there is no sunlight at all, and there's not very much for the rest of it. So, you know, when you're given lemons, make lemonade, okay, whatever it is. Okay, so in winter, we take healthy volunteers, we take blood from them, we load them up with 4 weeks of vitamin D orally. We take bloods again. So we start with people with low vitamin D, low sunlight. We take them to high vitamin D, low sunlight, because we need to show that there are UV effects which are not vitamin D. We then shine daily UV at them for 2 weeks. Basically, we use old-fashioned tanning lamps. So these are the closest thing to sunlight UV aspects, 90% UVA, 95%, 5% UVB broadly. We give them daily UV for 2 weeks, and we then repeat the bloods, and we also do skin biopsies pre and post UV. And what we can then do is we can do single cell sequencing. So there's this technique for the non-biologist here, this incredible technique now, where this blood, what you do, each blood test costs £3,000. I mean, it's unbelievably expensive. But in every single cell, so you take a blood test and you take all of the immune cells in the blood, the circulating cells, and in every single one of 10,000 cells, you calculate the expression of all of the genes, 30,000 genes, expression of 30,000 genes in 10,000 different cells, pre and post UV. Unbelievable informatics. Unbelievable amounts of data. You can make it cheaper by having male-female pairs, because men and women are different and they've got different genetic markers. And so you actually can combine a man and woman and you can get it to separate male cells from female. So you can halve the price of your experiment as long as you get male-female pairs. So what we're now doing is we're combining 3 lots of data. So we have our UK Biobank epidemiology population data. And in that we have disease outcomes, you know, incidence, mortality, cancer, cardiovascular, metabolic. There are also, and for every person, every one of those 500,000 people in the UK Biobank, we now have proteomic. There's now a protein score for the amounts of 3,000 different proteins in them, in their circulation. We then have mechanistic studies in man, where so in the biobank, we have a sunlight score attached to them based on our sunbeam score. In our human volunteers, we give UV. So we have a pre and post UV. We do the same proteomic measurements as we have in the biobank cohort. We also do single cell sequencing, looking at all the gene transcripts in circulating immune cells, and we do spatial transcriptomics, all the genes turned on and off in the blood. And I am then collaborating with Amaya Virus, who works down at the Cancer Research UK labs in Manchester. Amaya, like me, is a dermatologist. Like me, she's a dermatologist who doesn't go to dermatology meetings because no one wants to hear from us. She shines UV at mice. She then injects them with cancers. And what she finds is that the mice who've been irradiated have less cancer deaths than the unirradiated mice. She puts in melanoma and bowel cancer. In just the same way that our biobank people who've had more sunlight have less cancer deaths than those who've had less sunlight. We then do single cell sequencing of all the immune cells in the irradiated mice in parallel with the spatial transcriptomics, the single cell sequencing of the humans who've had it. And we find the same immune signature. And I can't tell you what this signature is. I'm sorry, because Cancer Research UK and University of Edinburgh patent lawyers are all over this, and we're about to submit what's going to be a major paper describing this. So we have the same immune signature in irradiated mice as irradiated volunteers. We have the same outcomes in terms of reduced cancer deaths in irradiated mice as the more sunlight human samples. We then have biopsy samples from people who've had cancer, and people who've had cancer and show this UV-induced signature have a better prognosis than people who don't have this immune signature. So it all hangs together as a really important UV-induced immune pathway that we've discovered that reduces cancer deaths. And of course, we know in the past that people in sunnier countries and higher vitamin D levels have less cancer deaths. And we know that giving them vitamin D has no effect. We now think we've got the mechanism. The other thing, doing cross-validation. So we are cross-validating. So we are trying to match up our experimental pathway where we shine UV at people with our biobank, people who've naturally had more sunlight. So we've now got a validation technique for showing that what we do to people matches up to the biobank. And what that is this incredible, I was saying that they took blood from in the biobank 15 years ago, knowing that technologies would be invented. Well, one of these amazing technologies first described in Nature Methods 5 years ago, so brand new technique, is this thing called O-link proteomics. Again, incredibly expensive, unbelievably powerful. So with a single blood sample, a tiny bit of a blood sample, 20 microliters or something, you can measure 3,000 different proteins, mind-boggling. And the way it's done is you have little antibodies to proteins, a pair of antibodies to a protein that are linked to oligos, to DNA tails. And so you put this antibody mix with antibodies to 3,000 different proteins into your blood sample. When those antibodies find a protein that they're targeted for, they bind on, and they have a unique tail on them, a DNA tail. And that DNA tail is then used to set off next generation sequencing, genetics, gene amplification. So you tie it to the existing workhorse of gene amplification. I examined a PhD, a very good PhD, last week in London, somebody who spent 3 years looking for protein signals and psoriasis outcomes, 12 proteins. So he's done a meta-analysis of all the papers studied, 12 proteins in history have been studied in protein. He spent 3 years looking at this. We now measure 3,000 proteins in a single blood test, straight off. Unbelievable. So what we do is so in the UK Biobank, we know when every single blood sample was taken and where it was taken. And what we can do is we can look at people in the UK Biobank whose blood was taken after 2 weeks of sunny weather. And we can compare them with people whose blood was taken after 2 weeks when it wasn't sunny. and we compare people whose blood was taken on a Monday with people whose blood was taken on a Monday and a Tuesday, because some people do different things. You know, they go for a run on the weekend, or, you know, they work. So behavioral things can affect it. So we do all the Monday people, all the Tuesday people. But we look at those bloods in people whose blood was taken after 2 weeks of sunshine versus people after 2 weeks of not sunshine. We then run exactly the same panel in our experimental subjects after 2 weeks of sunshine. And what we show is that they incredibly closely match up. So what we show, unbelievably close, what it does to protein expression. So what this means is that we know that that observational data in the UK Biobank for how much sunlight people get based on our scores exactly matches up to what happens when you give people sunlight. So we have a cross-validation. We know that the biobank represents what happens when we give people UV, and when we give people UV, we know it represents what actually happens in real life. So we have this only came out last week, really exciting stuff. So we've got really robust technologies here. So mechanisms of UV independent of vitamin D. Look, final bit, I'm dragging on. Skin color determines response to UV, as I showed with our studies on blood pressure. And look, serum vitamin D is a great measure for the biological effects of UV. And if you've got dark skin, you get less of a rise in vitamin D with sunlight. If you have dark skin, you do not get UV-induced skin cancer. I work in Ethiopia. I've never seen UV-induced melanoma. There are non-UV-induced melanomas, this rare type, not caused by UV, but UV-induced skin cancer only occurs in white Europeans. It doesn't occur in anyone else. And yet, this is the American and the British guidance on sun protection. We're going to be equal. Everyone's going to be on it. No, that's not being equal. You know, that's making it worse. Absolute madness. We all come from Africa. Those of us who are not recent Africans are descendants of the 1,000 or so people who left 70,000 years ago and scattered around the world. Humans have been in Europe 45,000 years. They've been in China 45,000 years. White skin develops independently in people who move to high latitude, low light environments. So the genetic variants leading to white skin in China are different from the genetic variants leading to white skin in Europe. So Chinese white skin, pale skin, arose around the Yellow River Valley. European pale skin arose around the Fertile Crescent. What is really interesting is how incredibly recently this happened. So Europeans in China and Europe 45,000 years, and yet pale skin in Europe only starts to rise about 8,000 years ago, and in China starts to rise about 11,000 years ago. So there's the cave art in the Dordogne. If you go to the amazing caves in Dordogne, that's 30,000 years old. The people that painted that had brown skin and blue eyes. Cheddar Man, earliest known Britain, 10,000 years ago, brown skin, blue eyes. There was a mini ice age called the Lesser Dryas 10,000 years ago, and he's one of the first people who repopulated subarctic Britain. See, the Iceman. You remember that chap who was found in that glacier on the Austrian-Italian border? About 20 years ago, they found this mummy there, frozen in the ice. Ötzi, 5,000 years ago. This is Ötzi. There's now a museum around him in Bolzano, in the Austrian-Italian border. That's Ötzi. They built a whole museum around him, and they'd done a life-size mock-up of him here. You know, he was going over a glacier. They gene sequenced him last year, published in Science. The reason Ötzi has brown skin, wait for it, is because he has brown skin. So, and yet, there he is with brown skin. There's the mock-up. So white skin is really, really recent, despite the length of time for which humans have been here. So what could account for it? So what is really interesting is white skin develops in China and in Europe when we start farming, when we undergo the Neolithic transition. So Fertile Crescent, 8,000 years ago, it's kind of wheat, sheep, cattle. Yellow River Valley in China, it's millet. Rice comes 1,000 years later, and pigs. So it's when humans start farming in a low light environment that pale skin is favored. So farming has arisen all around the world in different places. Humans have been a high latitude for tens of thousands of years. But when you get the two together, that is when there is an evolutionary fitness benefit for pale skin. Why is this? Could be dark, could be vitamin D. I think probably infection. And the reason for this is most infectious disease is zoonotic, comes from animals. And when you're farming, you're living by your animals, and also you're living in communities. You're living with other people, so disease can spread, rather than dispersed hunter-gatherer bands. So the last 100 years, no signal for infection. We've had vaccines, antibiotics, sewers, clean water, all of that stuff, removing the data signal. However, COVID came along, and we published a paper. We were able to look at COVID deaths and sunlight. So we started off America releasing COVID data at county level. So it's 50 states, 3,000 counties. And we looked at deaths back. So if you're going to die of COVID, you need to do several things need to happen. You have to meet somebody with COVID. You have to catch it. You have to get sick, fail to respond, and die. So you have to put in lots of corrections. So your chances of meeting somebody with COVID are based on population density. If you're living in Manhattan, it's very different from living in North Dakota. So you look at population density and percentage of the population with COVID. You then look at your chances of getting sick. So that depends on age, ethnicity, air pollution, poverty, or you then adjust for all of those things. And you throw a final random third factor in at state level, a random effect. Some states believed in masks, some didn't, some, all of that sort of stuff in America. You throw it in, and this is the first 3 months of the pandemic. One type of COVID, no immunity, no vaccines, everybody naive to it. More UV, less COVID deaths. Repeated in England, separate study because collect data differently. Same thing, more UV, less COVID deaths. Italy, more UV, less COVID deaths. And then interestingly, going back to the population data, reduced TB deaths, reduced influenza deaths, dose dependently, a mass of these diseases, viral diseases, less deaths, no effect on these bacterial deaths. It's not everything. But I think we're now starting to look at skin color in this. It suggests to me that maybe that driver to white skin has been infectious disease. Anyway, work in progress. And finally, we copy our Australian advice from Australia. So this handsome young fellow on the left is me 30 years ago as a medical registrar in Cairns, where they teach slip, slap, slop, the advice we're given in Britain. This is the UV index in Cairns. So midwinter, the UV index in Cairns is 7 for about an hour in the middle of the day. Midsummer, it's 14. So we have copied Australian sunlight advice, as Alex was saying at the start, 11 till 3. So that's where the advice comes from. In Scotland, we measured the UV index in Glasgow. So 2 years ago, the UV index in Glasgow hit 7 for 20 minutes on the 24th of June. 20 minutes all year. That was a sunny year. Last year, the UV index hit 7 for 5 minutes, also on the 24th of June. 24th of June, a great day to visit Scotland. So for 5 minutes in Scotland, we have the midwinter level of UV on Australia, and we offer Australian sunlight advice. Total madness. We are worried about skin cancers down here. These are deaths and disease in the UK. Melanoma right at the bottom. Sunlight reduces all these causes of deaths. It is madness. So, sorry, I've gone over my time.